Have you ever wondered why cardiovascular diseases such as stroke and myocardial infarction are the world's leading causes of mortality, accounting for over 18 million deaths a year? This staggering statistic underscores the critical importance of understanding and effectively treating these diseases. Cardiovascular diseases, a collective term for conditions that affect the heart and blood vessels, have a profound impact on global health. They often result from occlusions or blockages in the blood vessels that compromise oxygen delivery, leading to cell death in the heart or brain, and ultimately, stroke or heart attack. Our bodies are intricate networks of blood vessels, tasked with the crucial role of distributing blood across our organs. This ensures that active cells receive the oxygen and nutrients they need to function optimally and keep vital processes, like our heartbeat and brain activities, running smoothly. However, when these blood vessels are compromised, it can lead to ischemic cardiovascular diseases. These are diseases caused by reduced or absent blood flow, and they represent a significant challenge in the medical world. One of the key clinical questions that remain unsolved is how to effectively revascularize or restore blood flow to the affected organs. Each organ in our body fulfills a different physiological function, and so the vascular branching patterns differ across organs. This has long been a mystery in the field of cardiovascular research. How do these unique organ-specific vascular structures develop? Understanding this process from a therapeutic standpoint could open doors for developing personalized medicine strategies. These could be pivotal in combating not just cardiovascular diseases, but also neurodegenerative diseases and cancer. With such a significant impact on global health, the quest for new treatments and understanding of these diseases is of utmost importance. We may be standing on the edge of a breakthrough thanks to a team of researchers who have discovered a new cell type in blood vessels responsible for vascular growth. This revelation could pave the way to innovative therapeutic strategies to treat these life-threatening diseases. Stay tuned to learn more about this exciting discovery and its implications for the future of cardiovascular disease treatment. In a breakthrough discovery, a team of researchers at KIT have identified a new cell type in blood vessels responsible for vascular growth. This cell type, christened the endothelial L-tip cell, or more colloquially, the pioneer cell, has been found to play a crucial role in the formation of organ-dependent vascular structures. Now, you might be wondering, what makes this cell so special? Well, the answer lies in its unique movement. Unlike other cells, pioneer cells reside within the inner layer of our blood vessels, known as the endothelium. They have the ability to move inside the vessel walls, a trait that sets them apart. This movement is not random or aimless, mind you. Instead, it's very much like a dance orchestrated by signals produced by cells in the surrounding organ. Once these pioneer cells come into contact with these specific signals, a fascinating process is set into motion. The pioneer cells begin to form new blood vessels. To unravel the mystery of which cells produce these signals and how these signals promote the differentiation of pioneer cells, the scientists turn to a relatively new technique called single-cell sequencing. This cutting-edge technology combines detailed RNA sequencing of individual cells with bioinformatics analyses, allowing for precise identification of cell subtypes and the molecules these cells produce for cell-to-cell communication. With the help of single-cell sequencing, the researchers discovered that the vascular patterning is encoded by a distinct set of molecules. These molecules can only be sensed by a subset of endothelial cells, leading to vessel growth. In essence, the cells in the tissue produce an organ-specific set of molecules that encode the blueprint for creating a new blood vessel at a specific location and time. The identification of this pioneer cell opens up a whole new realm of possibilities in the treatment of cardiovascular diseases. This discovery could potentially pave the way for novel therapeutic strategies to treat ischemic cardiovascular diseases, ultimately saving millions of lives worldwide. But what exactly do these pioneer cells do? Let's take a closer look. Pioneer cells, or endothelial L-tip cells as they're scientifically known, reside within the inner layer of the blood vasculature, an area known as the endothelium. These cells aren't just sitting around though, they're active and dynamic, moving around inside the vessel wall. Imagine them as explorers navigating the complex terrain of our vascular system. As they journey, they seek out specific signals like a kind of biological Morse code. These signals aren't random. They're produced by cells in the surrounding organ, each signal unique to the organ in which it originates. It's like a conversation, a dialogue between the pioneer cells and the organ. The organ sends out these specific signals, and the pioneer cells, ever vigilant, pick them up. Once they come into contact with these signals, something remarkable happens. The pioneer cells spring into action and start to create new blood vessels. Now, you might be wondering, why is this important? Well, the formation of new blood vessels, or vascularization, is crucial for maintaining the health and function of our organs. When blood vessels become blocked or damaged, it can lead to serious health conditions such as stroke or heart attack. By creating new blood vessels, the pioneer cells are essentially helping to restore and maintain the flow of blood, oxygen, and nutrients to our organs. But it's not just the creation of new blood vessels that's important here. The interaction between the pioneer cells and the organ-specific signals also contributes to the unique vascular branching patterns seen in different organs. These patterns are not arbitrary, they're essential for the specific function that each organ carries out. In essence, the pioneer cells are the architects of our vascular system, shaping and building it in response to the needs of our body. They are the unsung heroes, working behind the scenes to keep our bodies healthy and functioning. The ability of these pioneer cells to initiate vascular growth is a significant discovery in the field of cardiovascular research. So, how do these cells know where and when to form new blood vessels, you may ask? Well, the answer lies in the cutting-edge field of single-cell sequencing, a technique that combines detailed RNA sequencing of individual cells with bioinformatic analyses. This technique allows for precise identification of cell subtypes and the molecules these cells produce for cell-to-cell communication. It's through this innovative method that scientists were able to discover a unique set of molecules that play a vital role in vascular growth. Think of these molecules as a sort of molecular cocktail. Each organ in our body produces its own unique blend of these molecules, encoding instructions on how to make a new blood vessel at that particular place and time. It's this molecular cocktail that pioneer cells are specially equipped to sense and decode. Much like a bartender who knows how to mix the perfect drink, the pioneer cells have the ability to understand the unique blend of ingredients in each molecular cocktail. Once they sense this cocktail, they are able to decipher the encoded instructions and initiate the process of vascular growth. But it's not just any cell that can do this. Only a subset of endothelial cells have this capability. These cells, already residing in the inner layer that lines the blood vasculature, are uniquely positioned to sense these signals and respond by promoting vessel growth. The discovery of this molecular cocktail and the specialized cells that can decode it is a major breakthrough in our understanding of vascular growth. It provides insight into how our body regulates the formation of new blood vessels and how this process can be influenced and controlled. This molecular cocktail is not just a bunch of random molecules, it's a precise organ-specific set of instructions that guide the formation of new blood vessels. Understanding this cocktail and the cells that can decode it opens up new possibilities in the treatment of cardiovascular and neurodegenerative diseases. This molecular cocktail is the key to understanding how our body regulates the formation of new blood vessels. What does this mean for the future of cardiovascular disease treatment? Well, this groundbreaking discovery of the endothelial L-tip cell, or the pioneer cell as we affectionately call it, has the potential to reshape our approach to treating cardiovascular diseases. Imagine a world where we don't merely treat the symptoms of these diseases, but address the root cause. By understanding the organ-specific molecular control of vascular growth and patterning, we could develop personalized medicine strategies. These strategies would be tailored to the individual, taking into account the unique vascular structures of their organs. For instance, consider ischemic cardiovascular diseases caused by reduced or absent blood flow. These conditions often lead to stroke or heart attack due to the occlusion of blood vessels and compromising oxygen delivery. With the knowledge of how pioneer cells function, we could potentially restore vascular perfusion and promote tissue regeneration, effectively revascularizing organs. Moreover, this discovery isn't just limited to the realm of cardiovascular diseases, it carries implications for neurodegenerative diseases and cancer as well. By understanding the organ-specific molecular control of vascular growth, we could potentially develop new strategies to combat these conditions. But perhaps the most exciting aspect of this discovery is its potential to enable the development of a molecular cocktail. This cocktail would consist of organ-specific molecules that instruct the formation of new blood vessels at a specific time and place. Once a prospective pioneer cell senses and unravels this specific tissue-derived molecular code, it could initiate the vascular growth process. These are early days yet, and there's a long road of research ahead. But the potential applications of this discovery are immense, and the implications far-reaching. It's a thrilling time to be involved in the field of cardiovascular research. This discovery has laid the foundations for revolutionary approaches in the treatment of cardiovascular diseases, bringing hope to millions affected worldwide.